I listened to four 15-minute presentations on April 5th, but I will only be discussing three. The first presentation was of mice in museums, utilizing natural history collections to investigate the relationship between cranial morphology and habitat, by Brandy A. Kraft. Their focus was on Peromyscus maniculatus, a rodent native to North America and a wide variety of environments, from forests that are tropical to deserts. Because their distribution is so diverse, Kraft wanted to see if there were any modifications to their cranial morphology, and they hypothesized that there would be modifications based off their environments. My main question is why there would even be any modifications, but somebody else asked first. Kraft thought that different diets, like harder nuts or softer vegetation, would encourage or discourage the enforcement of certain muscle attachment points on the skull. It made sense because, as explained in class, things like phenotypic plasticity might cause a regular eastern deer mouse to reinforce its bones to better break open hard nuts native to its habitat. For the experiment, they used 20 individuals, forested and non-forested, from various museums, CT scanned the fluid-preserved specimen, segmented out the individual bones, landmarked the right side of the skull, and plotted out the data. There was not a lot of clumping on their PCS plot, but when they calculated the differences, there was a margin of 0.076. Only two areas seemed to have been modified. The rostral extended, and the caudal zygomatic arch shifted down. However, they noticed a correlation with the habitat, but not causation. Their theory was that the differences could have been from possible cryptic species in the collections, subspecies, latitude and elevation, or it could have actually been caused by the diet, but they really don't know. I was honestly surprised that there was no causation, since adaptation is such a big thing. The second presentation lined up with the first. It was comparative shape differentiation in the forelimb and hindlimb morphology across habitats of the Peromyscus maniculatus species complex by Haley and Ellis. I did not manage to catch if they were an undergraduate or graduate student. Ellis wanted to focus on the same species as the previous presenter, and even use the exact same museum samples. They used the 20 North American deer mouse samples, 10 forested, 10 non-forested, to hypothesize if there was going to be some kind of difference between the habitats, and how they would influence the landmarks on the deer mouse's forelimb and hindlimbs. Since it is not likely to be influenced by diet, I wanted to know why there was any kind of modifications at all. So I asked, and Ellis said depending on if the mouse was from a flatter or more forested habitat, they might have to make certain muscles stronger to climb trees or run faster. To perform the experiment, they did the exact same process as the first presenter. The only data that ended up clustering happened for the femur, while the humerus and scapula had none. The femur was also the only one determined habitat variable, with an ANOVA rating of 001. The affected parts were the third trochanter, which became more robust and larger, and the femoral head, which became more round. Ellis said the results were inconclusive, and if they had more samples, they might be able to fully determine if the habitat did have an effect on anything. Again, connecting this to phenotypic plasticity and Wolff's Law, and the modifications that organisms go through even when they're the same species, I found both presentations interesting, since it was cool to see the possible modifications directly happening to the body of an individual due to their habitat. I was interested to see if similar effects could happen to people, since it's now very easy to communicate and record that kind of thing. For example, what were the differences between a skeleton of somebody living in a mostly hilly or mountainous area versus somebody living in a flatter landscape would be? Or somebody who eats mostly crunchy or harder to chew foods like jerky all the time would look like versus somebody that only eats softer foods like fruits and yogurt. Would that same kind of skull modification happen? I don't know. Would the first group have more defined chewing attachment points? Would the second group have weaker ones? That kind of deal. The final presentation was completely unrelated to the deer mice. It was triatomine vector host preference in experimental cafeterias, the impact of Trypanosoma cruzi infections by Jordan Solomon. Triatomines, otherwise known as kissing bugs, are generous vectors that feed on the blood of many types of hosts. However, they do seem to prefer dogs, chickens, and humans. Solomon hypothesized that the Trypanosoma cruzi parasite modified the kissing bug's preference for hosts just down to dogs, since chickens cannot transmit the parasite and humans are less likely to eat them. They conducted a live study with volunteers of dogs, chickens, and humans contained in 2 boxes connected by a long, see-through tube. They collected 188 kissing bugs, were starved for 119 days, so they would be more willing to find a host, and separated based on if they were infected or not. 102 parasites were parasite negative, while as 86 were parasite positive. During the experiment, the bugs reacted differently, with some choosing a host, some wandering the tube aimlessly, and others refused to move at all. When comparing the reactions of the ones that did pick hosts, Solomon noticed the uninfected chose dogs most often, while the infected refused to choose a host. That is, in the end, their hypothesis was incorrect. My question is why the parasites even didn't approach a host at all. If you're a parasite, you would want to find a new host to pass on, right? But Solomon believed this was because the Trypanosoma cruzi is more likely to be spread through consumption. So having the kissing bug wander around vulnerable would increase its chance of being eaten and the parasite passing on. Connecting this information back to class, the relationship between kissing bugs and Trypanosoma cruzi is only beneficial on one side and detrimental to the kissing bug. It also shows the evolution of parasites to influence an organism's behavior. I thought it was interesting because if this continues, will kissing bugs mutate and possibly build up an immunity to the parasite? I also just thought it was interesting since the effect that Solomon had desired to see reflected in parasite-positive individuals was actually reflected in parasite-negative individuals. Additionally, it's very scary to think what would happen since a parasite could directly modify an organism's behavior. I was wondering what kind of effects that could have if the infection jumped to people or different species besides insects.